Okay, hello everyone. Good afternoon from New York. I know it's late in most time zones where high TB burden countries are located. So thanks for those of you who are in time zones ahead of us for staying on and joining us. I think we'll go ahead and get started today with our Community Connect event, researchers sharing with communities results of a phase 3 TB treatment shortening clinical trial, which was presented yesterday at the Maine Union Conference. I am really happy we have the chance to discuss these trial results with the TB community so quickly after they were first presented scientifically. I'm Mike Frick. I work for Treatment Action Group in New York, where I co-direct the TB project. I'm also the secretariat of the Community Research Advisors Group, which is the community advisory board to the TB Trials Consortium, or the TBTC, which was the global research network housed at CDC that led this clinical trial in conjunction and close collaboration with the AIDS Clinical Trials Group at the US NIH. So we're going to talk about results of this study with our awesome panel today, which includes TBTC and ACTG researchers, members of the Community Advisory Board, TB activists. Before we do, I want to say just a bit in terms of introduction and how today's session is going to run, and then we'll get to it. I think we all know that for years, communities have been calling for shorter TB treatment. 6 months standard therapy is highly effective, but just too long. And the long duration creates all kinds of complications and difficulties for people with TB, their families, their healthcare providers, and health systems. And shortening TB treatment has also long been a scientific goal. And with the results of TBTC Study 31, ACTG 5349, it looks like we've finally achieved that goal and maybe have a 4-month TB treatment regimen that is just as effective at curing drug-susceptible TB as the 6-month standard we all know well. So before introducing the panel, I want to share a little bit of the language and words I was hearing yesterday after the results were presented and people were reacting to them. I heard several people call this study a landmark TB trial, a major milestone in TB treatment history, a signature scientific achievement. Some viewers of yesterday's presentation of results mentioned in the live chat that they had tears in their eyes as they were watching the results be presented. And for myself, I had a huge smile on my face the entire time. So clearly there's a lot for us to unpack scientifically, perhaps emotionally, and that's what we're going to do today. So this is really meant to be a chance for TB-affected communities, members of civil society, and the clinical community too, because I see we have lots of attendees here who are practicing TB physicians or work for donors or other health agencies to ask questions about the trial and its results. For our purposes today, no question is too basic. We really want this to be our chance to get all of our questions answered as soon as possible, and also to talk about what comes next for this 4-month regimen. So here's how today's session is going to work. In a minute, I will ask each of our panelists to introduce him or herself, do a quick self-introduction. And then we're going to run this as a question and answer where I'm moderating and asking each of our panelists some questions. We don't have a lot of slides to share. We may occasionally share some data with you if we need to talk about something specific, but we figured at this point in the week, we might all appreciate a break from PowerPoint, and it'd be better to keep the focus on the conversation. So it's really meant to be very interactive. And as I get the conversation going with the questions we planned, I really hope people put into the chat questions that you have. I'll be monitoring the chat and we'll be asking those questions of the panelists as well. And we have about 90 minutes ahead of us to get everything sorted. So we'll see if that's enough time. I'm sure questions will beget more questions, as usually happens. So without any further ado, I think we should get to it. Our first panelist is Dr. Payam Nahid. Payam, do you want to unmute yourself and introduce yourself?
Sure. Thanks, Mike. And thank you for organizing this. This is really amazing venue, and I look forward to our next 90 minutes. My name is Payam Nahid. I'm a professor of medicine in pulmonary and critical care in San Francisco at University of California, San Francisco. And I served as protocol co-chair for Study 31, along with Dick Chason, who's on, Susan Dorman, and Sue Swindells. And I also work with the Vietnam NTP and served as a co-lead with the National TB program there as an enrollment site for this trial in Hanoi. Thank you.
Thanks, Payam. Dick, you're up next.
Thank you, Mike. Good afternoon, good evening, everybody. I don't think good morning anybody, I hope. Nice to be here and exciting to talk about this. So my name is Dick Chason. I'm at Johns Hopkins University. I'm the director of the Center for TB Research, as well as our Center for AIDS Research. And as Payam said, I'm one of the co-chairs. I have worked with the TB Trials Consortium since its inception in 1994. And also was the founding chair of the TB Transformative Science Group of the AIDS Clinical Trials Group. So had a role in bringing together these two networks to conduct the trial. And I work with colleagues in Soweto, South Africa at the Perinatal HIV Research Unit, which is a TBTC trial site, as well as an ACTG site. Both of which participated in this trial.
Thanks, Dick. Next, Dorothy, how about you introduce yourself?
Okay. Hi, everyone. Good afternoon, good evening. It is around 9 o'clock in Uganda, 9 p.m., but I'm happy to be on the call. My name is Dorothy Namtamba. I work with the International Community of Women Living with HIV Eastern Africa, but I'm a member of the Community Research Advisory Group and attached to Study Site 30, which is one of the study sites that participated in this specific study, and a co-chair of the CRAG, the Community Advisory Research Advisory Group of the TBTC. Thank you.
Thanks, Doro. Glad you're here with us. Barbara, let's go to you next.
Hi, I'm Barbara Seaworth. I'm an infectious disease physician, and I have been taking care of TB patients for probably about 35 years. And I've been a member of the CRAG probably for about 10 years or so, and a co-chair for the last 3 or 4 years along with Dora. And I'm just really excited about the results of this trial. It's so good for patients.
Hey, Barbara. And then finally, we have Cynthia. Cynthia, do you want to introduce yourself?
Yes. Hi, everybody. My name is Cynthia. I am based out of New York City. I'm originally from Zambia, been in the US almost 20 years now, and I'm one of the longest serving CRAG members, just very passionate about TB work. Coming from Sub-Saharan Africa, you know, we have the highest incident and burden of TB. So really excited about these results, especially having known a lot of family members, friends that have had TB and have had to endure long treatment. So this is really great news and very, very exciting. Thank you.
Great. Thank you all for being here and for making the time. I think this is a really important event for us. And we should just get to it. So I'll ask the first question, and before we dive right into the results, which are so exciting, just to make sure we're all on the same page, and in case anyone didn't see the presentation yesterday, maybe we should talk a bit about the study design and its objectives. So Payam, the first question is for you, and it's just to ask you to describe the design of this clinical trial and its hypothesis. What drug regimens were you studying? What main question were you and the other investigators trying to answer with this study?
Yeah, thanks, Mike. The clinical trial tested 2 experimental regimens, both of which were 4 months long, both of which were built on a foundation of rifapentine instead of rifampin or rifampicin, which is the other name. And with the rifapentine having undergone extensive earlier stages of development in terms of getting the optimal dose. The drugs were administered daily, 7 days a week. The rifapentine was administered at 1,200 milligrams flat dose. And in one of the 2 experimental regimens, moxifloxacin was put in place of the normal ethambutol to add the additional potency of moxifloxacin. And the moxifloxacin was then continued for the full 4 months. So both regimens, rifapentine-based, optimized doses, one of the two with moxifloxacin, and this was compared to the standard WHO 6-month regimen, also CDC standard. So the international standard of HRZE, intensive phase, and HR for a continuation phase. The other, the other aspects of the trial perhaps that are relevant for people to be aware of is that food affects rifampicin and rifapentine differently. And so the trial could not be blinded. It was open label because in fact, those who took rifampicin really were advised to take the therapies on a fasting on an empty stomach. Whereas in rifapentine, people were encouraged to eat something beforehand. Not a lot of specifics on what to eat for this phase 3 trial. It was meant to be pragmatic. And then those patients were followed for 18 months post-randomization to really capture their outcomes in terms of after completing treatment, were they cured, and then were they durably cured for the follow-up period when they were just being monitored.
That's great. Yeah, we have had some questions emailed to us already about the food, so we'll get there eventually. And I remember many conversations designing the study about whether to recommend a hard-boiled egg versus something else. So maybe we can relive that too with people. But Payam, I'm wondering, so if we can develop for the purposes of our conversation, like a shorthand for these two experimental regimens. I think in yesterday's presentation, the one with both rifapentine and moxifloxacin was kind of called the rifapentine moxifloxacin regimen, and the other was just rifapentine alone. Does that work for you for the rest of today?
Yes, that works fine. We're also going to continue using that language in the publications and other presentations. So yes. Yeah.
Great. Maybe one day we'll name it, but we'll use that for now. Okay, so in terms of the study design, I think one thing that's really important for our audience today to know is that communities had an opportunity to inform the study design before the trial started and to review the protocol at multiple points when it was being developed. And Cynthia, you were the protocol representative or the community representative to the protocol team, and Doro was also very involved in this. So Doro and Cynthia, I'm wondering if you can each share an example of how community feedback was incorporated into the final study design and what impact communities had on this study from the outset. And maybe Doro, let's start with you, and then we'll go to Cynthia.
Okay, thank you, Mike. Communities, especially from our participation as the Community Research Advisory Group, was very critical in this study. And I'll share with you some of the things that we did, and maybe I'll share one or two, and then Cynthia comes in. One was around participation of the people living with HIV in the study. And although it had already been included in the inclusion criteria, we had realized that one, the people living with HIV CD4 cell count had been limited to around 200, and yet we realized that there are people who could have also benefited from this study with CD4 count which were lower. So one of the things that we did advocate for and we felt was taken on was to lower much more that CD4 cell count to at least 100 milligrams, 100 cells per milligram, and then that also brought in more people who are living with HIV to participate in the study. And the other one was around the adolescents, because one of the things that we realized was that adolescents was a new area where people, especially researchers, have reservations in terms of participating in the study, that they are dynamic and probably they may be difficult. But as a CRAG, we advocated for the inclusion of progressive inclusion of the adolescents up to at least the age of 12 in the inclusion criteria. And this was something that was also taken up. I want to take it over to Cynthia.
Thanks, Doro. So yeah, it is, as Mike says, collaborating and ensuring that the needs of the community really met in the protocol team. They did include us, and CRAG played a very, very important role in ensuring that those needs were addressed and the protocol reflected that. So like Doro said, some of the things that we did add and made sure that people with cavitary TB were also included. Initially, that was being debated, but it ended up being included in the protocol, which I think, again, with people living with HIV and the CD4 of 100 and lowering it to 12, though we would have wanted to lower that age much lower, but at least, the researchers were able to allow the 12 years old, anybody 12 years and above, to be a part of the study. And for us, that was really a win, and it's just a step in the right direction for future research and ensuring vulnerable population and other populations that are otherwise never included can be included. And I think one of the things we'd like to see maybe in the future, building off of this research, is really even focusing on pregnant women and lactating mothers. So that wasn't included in here, but those are some of the things that we as a CRAG keep in mind.
Yeah, thank you both. And I think, Cynthia, that last point was really critical. Like, we hope this trial kind of sets a precedent for more inclusive inclusion criteria for future TB drug trials with respect to cavitary disease, age, HIV status, and some of these other things you guys mentioned.
Mike, could I just make a quick addition there, because this really is an important point to hammer in. In the long journey of the protocol development for Study 31, some of the most memorable moments in my memory were around when CRAG and the community engagement brought forward influential advice around adolescence, CD4 count, and we adopted it. We took it, we discussed it, and we included it in the protocol. And I won't ever forget that. I'm most grateful for it. I wish more regimen developers will do it, and I hope they will. I think they will. I think this is going to provide a sort of a new benchmark, if you will, from that perspective.
Yeah, amen. Okay, one more thing about study design that I want to talk about, and then we'll talk about results. And Dick, this one is for you. So I think an important detail of this trial is that it's what we call a non-inferiority trial. And as someone who's conducted other TB non-inferiority trials, I thought you would be the right person to tell us what this means and how we can best kind of understand non-inferiority and interpret this kind of counterintuitive concept.
Yeah, it is somewhat difficult to grasp sometimes, but let's start. I'm going to explain it just in lay terms, like I would explain it to my brothers and sisters, none of whom are researchers. Actually, one of them is, but so non-inferiority means it's as good. It's as good. But in the old days, we used to do equivalence trials to say that it's equivalent, meaning it's the same. It turns out that equivalence trials are hard to design because they require huge numbers of people to say that something is exactly the same. And then you have to argue over, well, what's the same? If this cures 98% of people and that cures 96% of people, is that the same? And so what's evolved over the last couple of decades and really become widespread in the last 10 to 15 years is non-inferiority trials, where you say it's not inferior, it's not worse, and you pick based on a number of factors what would be worse. And one important reason to do a non-inferiority trial is when you have a new treatment that has advantages that you would prefer, and you're willing to accept that it's not identical to the old regimen, the standard regimen. You're willing to accept that it's not quite the same, but you don't want it to be much worse. So you say, you pick a level. And in this case, we picked a level of 6.6%, that the estimate of what, the worst likelihood of the regimen being was 6.6% worse than the old standard regimen. And the reason is that we're willing to accept small differences in efficacy, effectiveness, for economies of, treatment, for easier treatment, for safer treatment. So if we can say that a 4-month regimen is non-inferior, it's not worse, it's as good as a 6-month regimen, even though it's not identical, it has so many benefits that it makes it worthwhile, and we accept that it's not identical. Now, it turns out, when we can get to the results, the actual results are very small. So that makes us even happier. We didn't come anywhere near that 6.6% difference with the winning regimen. But that's why it was done that way. We want to show that it's as good as, but we'll accept that it's not identical to or even better than, because it has so many advantages.
That's really clear. Okay, and so I think we should get to the results now. And so maybe Payam, do you want to summarize in terms of the efficacy analysis, how each of these two regimens, the rifapentine regimen and the rifapentine-moxi regimen, how they looked compared to control in relation to this non-inferiority concept Dick just introduced?
Sure. I have so many things I want to say, but I think Dick really described non-inferiority beautifully, to consider this test of whether a new regimen that's shorter, these two 4-month regimens, is as good as the 6-month regimen. And without showing slides, because we're going to try to avoid slides, the combination rifapentine-moxifloxacin regimen, the 4-month regimen, met this non-inferiority goal, this benchmark that Dick described. Emphatically, the results show that it was non-inferior to the standard 6-month regimen in all the population groups that were analyzed, in all the subgroups that were evaluated, in 14 sensitivity analyses, in an assessment of burden of disease, in other words, looking carefully at those who had the worst or the highest severity of disease burden at outset. It just was robust. It's a robust 4-month regimen and met that benchmark. And I think part of the reason why we're so happy, and this is a shared community achievement, frankly, because this has been a decades-long journey. This is not just the achievement of the TBTC or ACTG, it's a shared achievement for the TB community at large, is because we have, just for context for everyone to understand, the last time we were able to reduce the duration of TB treatment for drug-susceptible pulmonary TB was 1972, when the British Medical Research Council published early results of a 6-month regimen, streptomycin, isoniazid, rifampin, as being equal to an 18-month regimen that was the standard then. And we've been with a 6-month regimen since 1972, despite many, many trial attempts to get shorter. And this is the first regimen that has met accepted benchmarks and proving that it's as good as the 6-month regimen across the board, insofar as subgroups, analyses, etc. Now, the rifapentine alone regimen did very well as well, actually, but it didn't meet this specific benchmark that Dick described. But it was still able to cure But first of all, it was still able to cure 86% of the population that received that single substitution, the rifapentine instead of rifampin. 86% of people were durably cured. And in looking at it compared to some of the other regimens that have been tested in the last 10 years, 15 years, it's the second most potent drug regimen we've had to date as a 4-month regimen. But it didn't meet all the benchmarks that rifapentine moxifloxacin containing regimen did. And that was very clear.
So a kind of a divergent outcome for the two regimens. They both cured most people who received them, but the rifapentine moxi regimen did as well as, or was no worse than the standard based on this non-inferiority design that the trial set up. And it sounds like any way you sliced the data, in terms of looking at different patient populations within the trial, different sensitivity analyses that you did to test your findings, that was robust, a robust finding that held up.
Yes.
Right. Okay, great. So one, I think special area of concern for a lot of TB affected communities is, how well these regimens perform in people living with HIV. Who, as we heard from Doro and Cynthia, were included in this trial, people with HIV with CD4 count 100 and above. Dick, I'm wondering if you can kind of summarize like how the regimen, the rifapentine moxi regimen, looked in people living with HIV. Are there any special considerations for using this regimen in that group that communities need to be aware of or that we should be thinking about?
So first, let me explain about the inclusion of people with HIV in the study, because that was a point of discussion with the CRAG. I don't think there was ever any doubt, but what criteria would be used was debated. It was debated within the team and with the CRAG having input. And so we elected to go with a cut off of a CD4 count of 100, and that was based on an earlier ACTG study that showed with people with lower CD4 counts, there were so many AIDS-related complications in addition to TB that it would make it difficult to figure out, what's TB related here and what's HIV related. So that's why that cutoff was used. The other consideration is drug-drug interactions with antiretrovirals. And so when we started with the world using efavirenz as the standard of care at the time the study rolled out, we started saying that people who are already taking efavirenz can enroll in the study, and then we can evaluate the interaction and make sure it's safe, and then broaden the study, enroll people not taking efavirenz yet, people who would be going on efavirenz. And that reduced the number of people with HIV who got into the study, frankly. It was unfortunate, but it was done for safety concerns around the drug interaction. It turned out there's no drug interaction that matters. It didn't have any effect. So that's good to know going forward, that efavirenz can be easily used in people receiving this regimen. So that's the background about the people with HIV who were in the study. The result for the rifapentine moxi is it was better. Now, I'm saying that not in a statistical way. I'm just saying that if you look at the results, they did better. Their poor outcome, our treatment, unsuccessful treatment rate was almost double in the people who got the control regimen than the people with HIV who got the rifapentine moxi. They really did surprisingly better. Whereas for the rifapentine alone, the single rifapentine, they did much worse, people with HIV. So it was such a shocking and stark demarcation between the regimens. This regimen was great for people with HIV. And I can tell you, nobody expected that. And then I don't think any of us expected that the rifapentine regimen would be so much worse either. So again, I'm speaking not statistically here, because these are not statistical analyses. These are just, you look at the results. And for people with HIV, it was about 8% 8 to 9% had unsuccessful treatment with the rifapentine moxie versus 16% with the standard of care.
So it sounds like we should feel pretty good about using the rifapentine moxie regimen in people with HIV. We have had some questions come into our group by email overnight, though, about what about these newer antiretrovirals? Like everyone is sort of being transitioned to dolutegravir-based therapy. We looked at efavirenz and it was okay for efavirenz, but what about DTG? What about future ARVs, maybe like bictegravir, whatever else is coming down the pike? What do you think about those?
Yeah, so dolutegravir, I guess, is the most important, given that it's being rolled out worldwide. And what we know, well, I guess to begin with, what we know about the interaction of dolutegravir with TB drugs is that rifampin reduces dolutegravir levels enough so that it's worrisome that it doesn't maintain adequate levels to suppress HIV replication. And so a study done by my colleague Kelly Dooley looked at giving the dolutegravir twice daily. And if you give the dolutegravir twice daily with rifampin, it's very effective. That the twice daily dosing overcomes the drug interaction with rifampin. We expect that the drug interaction with rifapentine and rifampin will be very similar with dolutegravir. We've done studies of dolutegravir and rifapentine given for preventive therapy once a week, and they can be safely administered with standard dolutegravir once a day. But no one's done a study yet of dolutegravir with this daily regimen of rifapentine, either for treating TB like this study or for preventing TB like the brief TB study for prophylaxis. So those studies are getting done now. If I had to, suggest how to handle that at this point, I would say that using the twice daily dosing of dolutegravir would be the best bet until we have more data. But I think that in the end, that's what is likely to be the case. So I'll just add that with the rollout of dolutegravir around the world, what is being rolled out is a 3-drug single pill regimen, TLD, tenofovir, lamivudine, and dolutegravir. And because so many people have both active TB and HIV infection and need treatment, single dose dolutegravir tablets are also being distributed as part of this rollout so that people with TB and HIV can get treated with both. So they get the TLD for their HIV and they get a second dolutegravir dose in the evening to maintain adequate levels of dolutegravir while they're on their TB treatment. So that's already dealt with. The interesting thing is that the cost of that second dose of dolutegravir is less than the cost of the 3-HP regimen to prevent TB. But that's another point that I need the Craig and Mike to go to war over and get those prices down.
Yeah, we need to talk about price. We need to talk about price at the end here, or after we say a bit more about results. Let me. So we need some studies on this regimen and dolutegravir-based therapy. It sounds like, and we hope that maybe double dosing dolutegravir can overcome any kind of effect as it did for rifampicin. So we'll wait to see on that. So we have a couple of questions I think it'd be good to answer now. One person is asking about the gender balance of trial participants and how the trial looked in terms of enrolling men versus women, and whether we think this is relevant for how the regimen might perform. So that's one, and let me ask the second too, which is, this person is saying this result is really good news. Is the rifapentine moxie regimen, this shorter 4-month regimen, is it allowed to be shorter because of some kind of improved molecule, like the drugs are more powerful or more potent, or is it because of improved adherence by the patients? So maybe we can talk about if there was any difference in adherence between study arms. Anyone who would like to answer this question can go for it.
I'll take the gender question. It's a good one. I think our trial had 70% male self-identified participants. But this matches up with the global epidemiology of TB, and in virtually all countries around the world, there's more male than female cases of TB reported. Reasons for that aren't totally understood. The ratio is usually 2 to 1. And there could be biological reasons, socioeconomic, cultural barriers to access of healthcare. It's really a mix of things that nobody quite understands. But our sort of proportions enrolled 70% male, 30% thereabouts female, were equal to what the epidemiology is globally. So I hope that addresses that question. We didn't undertake any specific recruitment strategies to enrich, for example, if that was also a question, but we tried to follow the global kind of epidemiology.
So regarding the drugs, there were no differences in adherence, really. We looked and did analyses by adherence levels, and nothing stood out in terms of adherence because a lot of effort went into promoting adherence. The reason this regimen is better is it's just much more potent. So we know that rifapentine has 4 times the potency against TB as rifampin. It's 4 times as active. We also know that it lasts in the blood 4 times longer than rifampin. So you have 4 times more activity for 4 times longer. And we gave double the dose of the standard rifampin. So it's much, much more active. And then moxifloxacin is also a very active drug against TB. When you put them together, they're particularly potent. And we've had a couple of previous studies where moxifloxacin and rifapentine were put together, and it really improved the activity of the regimen. So it's a better regimen. It's not anything to do with adherence.
Great. So we've been talking mostly about the efficacy results. I think we should turn to safety, and I want to get Barbara involved here in a minute. But first, maybe Payam or Dick, do one of you want to kind of quickly summarize the safety profile of the rifapentine moxi regimen in comparison to the 6-month standard? And then after you do that, Barbara, I'm curious to hear from you, as someone who's been treating people with TB for over 3 decades, and as someone who I think is really keyed into issues of patient care and patient safety, how you view the safety results of this study. So first, Dick or Payam on the safety results, and then we'll go to Barbara.
I could give this sort of summary, and then Dick, please chime in. The summary of this is that there were no differences found between the experimental regimens and the control regimen in the primary safety outcome. So the trial itself was designed to capture a lot of safety and tolerability data. The analysis was focused on the proportion of patients who had experienced a grade 3 or higher adverse event. That's sort of a little bit technical language, but essentially, serious adverse events during study treatment. No difference. There were also no differences between our standard control arm and the experimental regimens in secondary safety outcomes, and these included tolerability, all-cause death during treatment, follow-up liver enzyme increases, and in some facets, in some of the areas of concern around safety, the rifapentine-containing regimens were better. They're just better. The rifapentine moxi was also better in several of the categories of adverse events than the control arm at 6 months long that we're all using. So it was really encouraging, and again, robust across the board.
Anything to add, Dick, to that?
No.
Okay.
Good.
So Barbara, then over to you. And as a practicing TB clinician, sort of what's your view of the safety results of the regimen?
I was pleased with the safety results. I didn't see anything there that would make me concerned about using this regimen on individual patients. And in fact, I think it's very encouraging and should be rolled out. Obviously, we'll need to do some continuing looks at how the regimen works in clinical practice, like was done with the 3-HP regimen, which I think will be helpful. But over the many years of using fluoroquinolones for a broad array of different TB-related type of patients, from drug resistant to those who don't tolerate erythromycin, or maybe just INH resistant disease, serious reactions with the fluoroquinolones have been really unusual. Obviously, some patients do have tolerability issues, and those can be identified and medication stopped. But I really don't recall any long-term serious irreversible reactions to fluoroquinolones in the patients I've treated over the years, and there have been hundreds and hundreds and hundreds. So I feel comfortable with using this regimen.
I want to talk about the fluoroquinolone issue a bit more. And so when we're talking about the fluoroquinolone, we're talking about the moxifloxacin, that drug in the regimen. We did get a question by email from a TB survivor expressing some concern because I guess the drug label for Moxi has some black box warnings on it. And so I'm wondering if Barbara, Dick, Payam, if you can comment on kind of what's behind that. What do we know about the safety profile of Moxi? We don't have to get into the weeds, but I think I've heard several people kind of ask about Moxi, and it's been interesting just in the 24 hours since the results were presented to see that, at least the questions I've been getting, nobody's been asking about high dose rifapentine. Everyone wants to know about moxifloxacin, which we've studied before for treatment shortening. So we actually have a lot of data on this, and I'm wondering if we can talk about it for a minute.
So maybe
I don't know who wants to start.
Yeah, maybe Payam could go first and talk about the experience of the TB Trial Consortium, or Dick, and their experience with using the drug, where they've really collected and looked in a systematic way for toxicity. And then certainly one of the rest of us can talk about what's in the literature or what patient complaints have been brought forward. I think might be a good way to think about it.
Sure. I'll just start globally by just reminding everyone that the warnings the FDA had released around these therapies were frankly focused on the use of the drug in settings in which other therapies were readily available, and there's an era in our use of the fluoroquinolone class of drugs where they were just handed out for any upper respiratory infection or urinary tract infection. And so the FDA's language anyway was, and I don't have it exactly in front of me, I'm paraphrasing, was basically for, those conditions, other therapies should be considered. We're talking about TB, we're talking about a deadly disease that we don't achieve the success rates that we have hoped for in the community, and there's a lot of suffering that goes on from the disease itself. So I think that's context that's really important in how to interpret these warnings that we get from regulators. I'll hand it to you, Dick, to expand.
Yeah, so two things to say. First, in the study, we didn't find any signals of problems with the fluoroquinolones in particular, in any of the things that people worry about with fluoroquinolones. But as Payam said, at the time of the anthrax attacks in the United States after 9/11, some surveys were done, and it was estimated that 50% of people in America had taken ciprofloxacin. Well, that's overuse of an antibiotic, and so it had to be dialed back to give them to people who really would benefit from them, and for which other alternatives weren't as good. A black box warning from the FDA after a drug is approved, like moxifloxacin or ciprofloxacin or any other drug, is a way to draw attention for the medical community to a newly described problem that's important enough that people should know about it. Doesn't it? And I think in everyone's mind, they think black box, isn't that what you do when the plane crashes? You get the black box. And so it has this image of, oh, it's really terrible. And a black box warning is a warning to bring things that doctors particularly should know about when they prescribe a drug. And so some of the side effects that have been reported with the fluoroquinolones are things that happen to 1 in hundreds of thousands or 1 in a million people. But there's an association, and the FDA wants you to know about it. But it doesn't mean you shouldn't use the drug. So that's the black box caveat, I guess. So in the case of the fluoroquinolones, for the things, I do have it open in front of me, Payam, and for the things that are listed, we didn't see any of them. Now, that's because they tend to occur very, very, very infrequently. But there's a risk with almost any drug that there could be a rare side effect. And we wouldn't want to suggest that no one who gets treated with amoxifloxacin will ever have one of these side effects, because they will. They do occur, but they're very rare.
So I think one person who had a concern about this was Arne von Delft, who has asked, he's raised his hand to speak. So Arne, I'm gonna try to unmute you. I hope it works. If not, apologies. But I think you're now allowed to talk if you want to go for it.
Yes, thank you. I don't want to create the wrong impression. I'm very excited that we are looking at a 4-month regimen and that we are looking at better options. I'm just careful about the language around safety, because fluoroquinolones are not safe. They've got 4 black box warnings on it. Speak from personal experience. I took it fluoroquinolone 2 types for 6 weeks. The side effects are severe and they are not uncommon, and I can speak through all of them. But I think the context is important because regular TB treatment is not safe either. The reported severe adverse events in all the trial arms is still higher than we would want. The success rate is still lower than we would want. So I think we need to be careful when we talk about fluoroquinolones being safe. They're not safe, and I will state that categorically. Whether they are less safe than the alternatives currently, that doesn't seem to be the case looking at the trial data. But as a civil society group and a TB community, we have to keep on pushing for safer drugs. Firstly, for TB preventive therapy, fluoroquinolones are definitely not a good option to treat people that are not sick, to try and prevent illness. But even in people that are ill with regular TB, and even if the side effects are rare, remember we have in excess of 10 million people with TB every year. You put 10 million people on fluoroquinolones, their tendons will rupture, they will get irreversible neuropathy, there will be severe mental health side effects. So I just wanted to put that out there. We have to keep on pushing for shorter and safer drug regimens, please. Thank you.
Not setting the safety issue aside, Arne, but I mean, I think part of what you're calling for is more research for even safer and better, more tolerable regimens. And I think no one says this is the end game and where we stop in terms of how to improve TB therapy. So that point is very well taken. We can keep talking about quinolone safety, and it's a conversation that will obviously continue past today's webinar. There's another aspect to it that I want to make sure we have time to voice. So let me raise it here because we've also received some questions in the chat that kind of touch on this issue. And then I had a question emailed to me by Zarir Udwadia of Hinduja Hospital in Mumbai, who pointed out that at his TB center, 40% of people with TB have pre-existing resistance to quinolones. And so what do we think about using this regimen in settings such as India, where there is a high rate of background quinolone resistance? What do we need to be talking about or advocating for as civil society in terms of drug susceptibility testing? What does a kind of diagnostic algorithm look like to put someone on this therapy? Who wants to handle it? it's a lot, I know, but Barbara, you look like you want to talk. Maybe we can go to you.
I believe that fluoroquinolone susceptibility should be up front with molecular testing for rifampin. I frequently will ask for, as soon as I get an expert that's positive, I frequently will ask for fluoroquinolone susceptibility in patients who I think are at risk of having fluoroquinolone resistance, or in whom I'm going to depend on a fluoroquinolone for part of their treatment. And we see that with the frequent use of fluoroquinolones, we are always popping up with one patient here, one patient there, and it's a small number, but for that patient, it makes a difference if you're going to base your regimen on a fluoroquinolone, and it turns out they've taken a ton of fluoroquinolone for urinary tract infection or for multiple pneumonias prior to the diagnosis of TB, and it's really important to know that, and you really can get the results quite quickly if you take the time to ask for them. CDC will do that testing if you ask for them with a real reason behind your ask. Same thing for upfront INH. So I know globally, that's also something I think that needs to be advocated for, especially in areas like Mumbai that have tremendous baseline fluoroquinolone resistance. Probably other areas of the globe where you can buy the fluoroquinolone on the street. There are other areas, Southeast Asia, that really need it as well.
So I'll add, glad Dr. Wadia asked that question, and I know Peter Sadowski asked the question about resistance in the chat that I wrote a quick answer to, in the Q&A rather, but I agree that we have to have rapid, accurate, available drug susceptibility testing for all people to use this regimen or any regimen. Until we have some fine day, the pan-TB regimen that treats everybody and no testing is necessary, we need drug susceptibility testing. It's critically important. In this study, the majority of people were diagnosed with expert. So they would have had a baseline rifampin resistance test. And if they had resistance, they weren't eligible. Then full drug susceptibility testing was performed after people were entered into the study. And about 5% were found to have drug resistance and couldn't be treated with the experimental regimens. And most of that drug resistance was isoniazid and some rifampin, very little of it, but some of it was fluoroquinolone. But it's essential to know that. And so we need to have first line, as close to point of care as possible, drug susceptibility testing for treating all patients with the experimental regimen or the control regimen. So I couldn't agree more.
Great. Okay, so DST is obviously one thing we have to advocate for if we're advocating for access to this regimen. Cynthia and Doro, coming back to you two, I want to start to think about kind of next steps and where we go from here, what the advocacy agenda looks like, how we communicate and convey these results to communities who aren't on the line with us today or may not watch the recording of this session. Where do you guys think we need to start? And what are some of your asks and hopes for, like, say, the next 6 months of sharing this clinical trial result? Cynthia, do you want to start this time? Yeah.
Yeah, so I think, I mean, it's great that we have these results, but also what does access look like? I don't know what, other countries, whether they've updated, they're going to update their national TB guidelines, and whether this regimen will be readily available and how long that will take. And also really looking, like this is just the first step, looking for ways to continue research and include other populations that have not been reflected in this study. So I think the number one is just access, making sure that this gets to the patients as quickly as possible.
Dora, what about your views?
Thank you, Mike. My view is, in addition to what Cynthia has just said, I feel that for us, from our perspective as communities, but also as advocates, probably work has just started on our side. That yes, it's about access, but we don't also know. I've just seen a post from the chat that WHO has welcomed the results with. I mean, it has really welcomed the results, and that is really a good sign. Most of our countries really look up to WHO and what it says around the different researches that are going on, but also to see how this is included in the new guidelines. So work has just started, but what we have already done, as the results were being released yesterday, we did posts to the different groups that we do exist, to the different communities, to our club members, to share with the communities that they can, that the results are very, very, very promising. And that what needs to be done also is that how do we engage at country level, that these results, yes, are there, but how do we engage with our NTLPs, that national TB programs, to see that, yes, personally, I did post to the National TB Program Manager, but at that level, yeah, that these are new results that are coming in, but how do we embrace them? And that they need to know this information is not, this research was not done anywhere else. It was done in Uganda, and it was done in many countries. So we need to benefit from such good results and such promising regimens. So our advocacy starts now. We need to see that whereas WHO is welcoming the results, that translates into new, guidelines, but also that also our countries are also responsive to what is coming out from what our research is doing. So we hope that we'll be engaging with our civil society, our communities, but also our national TB programs to ensure that they know about the results, but that also should be translating into access, because these are really things that are results that are promising in terms of eliminating TB in our communities.
Yeah, exactly. Okay, Dora, let me stay with you for a minute. So you already shared the results with your NTP manager in Uganda and
Yes.
with your community advisory board, which is awesome. And I think it's important for people to know that your site in Kampala enrolled, I think, 20% of the overall participants in this study. So like a fifth of the participants came from Uganda. We couldn't have done it without the Kampala site. So do you have any immediate feedback from your CAB members or from the NTP manager that you want to share with folks today?
Yes, from the NTP manager, the immediate reaction was like, these are very, very good and promising results. These are very good results. And that, yes, and the other comment that came in was that, yeah, but also we need to see how we also improve our diagnostics. But then I went back and said, yeah, of course, 40 years down the road, that took us that long to get to a 6-month regimen. But now that since the 1980s, when we had a shortened regimen to, shortening the regimen to 6 months, then you know how that pipeline is. Now that we have even a shorter regimen, we should not stay in the back and wait. But of course, she said these are really good and promising results, and that we hope to see how we engage more on this. That was what she did. And then from the CAB, they were really excited, and they were asking, we were really deliberating on what are our next steps. So we have had already a conversation with our site coordinator, Dr. Grace, and that we were asking how do we engage with other civil society or other communities, and that, yeah, so that the results are even known beyond what we are posting, that we have physical meetings. Say we could also involve other stakeholders in terms of even the NTLP, but also those funding agencies that we have paper in the house. I mean, in our countries, and that probably if we bring them to know, in case they, of course, I know they have known somewhere, but that we are disseminating, and that at country level we are sharing something that we need to move forward with. I think that would be helpful, but we have already talked about this with our study coordinator at Kampala.
That's great. I think it was really nice to see the email from WHO this morning saying that they acknowledged the trial results already and intend to trigger the process for a guideline development group and a review of the data when the final data are available. So hopefully we can move quickly and get these data reviewed. So some of the questions being posed in the chat are being answered in the chat by Dick. Thank you for that. So look for answers to some of these questions, which we're getting not verbally, but written. And Barbara, over to you for your views on this access and next steps situation.
So I love your enthusiasm for pushing hard for this, Doro. Listening to the presentation yesterday from Uganda, where I think it was so long for the standard 4-drug regimen to be adopted in Uganda by the National TB Program. You really don't have another 15 or 20 years to wait on this one. But I think it shouldn't be assumed that tomorrow this will be available in the US, and I bet it will take a bit of time. And I think we can shorten that time if there is a push from communities and clinicians to get this implemented into TB programs in the US. I know our center, the Heartland Center here in San Antonio, sent out a very wide blast to all of the nurses we work with across our region and many places in the country, to our medical consultants, to our state TB program managers. So hopefully we've already heard back from the TB program manager in Texas that she wants to get some of her physicians together and look at how we're going to implement this. So hopefully implementation will not take a year or two here in the US either. So I think we have to push across the world for this to be implemented, at the same time for implementation to continue to look at toxicity and how we might be able to expand it into other groups going forward.
It's not going to be easy in the United States, I think you're right, or not going to be immediate. We do have a head start in that rifapentine is registered here for the treatment of active TB disease. It's not everywhere else. So there are also a lot of other things that have to start to fall into place. It's not all on WHO or the NTP. It's also about registering rifapentine, manufacturing it, seeing what the cost of it looks like. Does anyone have any reflections kind of on that side of the access picture, the pricing situation, the affordability, the availability, the national drug regulation, and what needs to happen? Anyone who wants to chime in with thoughts on this is welcome to do so. I think a lot of it is still kind of unknown now. It's an agenda before us, but we can start to lay out what it looks like.
Mike, you and I both have experience in the last couple of years of working on getting rifapentine access in low and middle income countries, and it turns out to be much more difficult than I certainly ever thought. And when I compare it to what's happened with HIV drugs, it's almost night and day, and it requires sponsorship and advocacy and an entity taking responsibility for doing the filing to get the drugs approved, and that requires a lot of resources. So getting the drugs available in countries is a challenge that requires resources and effort to make happen. But it can happen, and we see that with the HIV drugs. Dolutegravir is available almost everywhere. It may not be rolled out everywhere yet, but that process has just gone so rapidly compared to rifapentine. So that really requires attention. In terms of price, both rifapentine and moxifloxacin are available from the Global Drug Facility, if you use their list prices, the cost of this regimen would come in at somewhere just under $200 for the regimen. And that is probably 3 to 4 times higher than the 6-month regimen is. And it probably varies by country because there's a range of prices. But the lower price would be somewhere in the neighborhood of $180 to $190 from the Global Drug Facility at their list prices. So that's likely going to require some negotiation to try to bring not with the Global Drug Facility, but with manufacturers, both the brand name manufacturers and generic manufacturers, to bring the prices down.
Thanks for that. So we've learned that some of the chat may not be visible to you all. It's visible to us as panelists. This is a setting on Zoom, which we don't control. So if there's someone from the union present, if you can change that setting for us so that the chat becomes visible to everybody, that would be great. We won't lose these comments that have been sent to us. We're going to collect them and we'll send them around, in a kind of write-up of the event. So don't feel as though we're censoring you. That wasn't the intention, and we're keeping track of it. So Dick, you anticipated an exercise which Sharon and Lynch of MSF told me we have to do right before the session, which was look at the GDF list prices for the drugs in this regimen and compare it to what we have for the 6-month standard and see how they shake out. And I hadn't done that yet, but you did the comparison for us, and it sounds like around $200, which very expensive compared to what TB programs are used to. I think another thing I want to flag for everyone to think about is the issue of pill burden, right? Because a lot of TB programs have been using fixed dose combinations of the 6-month standard regimen. And the 4 drugs in the rifapentine moxi regimen don't, as far as I know, exist together in a fixed dose combination the way that we have for first-line TB treatment. And so do we think that initially, until manufacturers respond, patients might actually have to swallow more pills for a 4-month regimen than they would for 6 months? Has anyone done the math on the trade-off between the duration and the pill burden based on the formulations? That's maybe another assignment for us if we haven't done that yet.
I don't think it requires math.
Good.
There are a lot more pills.
Yeah.
There are a lot more pills. If you're talking about the fixed dose Rifa4 and Rifamate combinations for the standard of care, this has more. The rifapentine moxi has more because of the rifapentine.
Okay.
Now,
so
even with the extra 2 months lopped off, I still think that obviously saves some, but I think what matters in pill burden is actually the daily pill burden. Every time you take this, you have a fistful, and that's a lot of medicine. And we clearly need better formulations. We clearly need fixed dose combinations, or at least rifapentine that has more drug per tablet than the current form.
On the chat, it looks like this may be resolved, and it may be up to the person who is sending a chat to choose whether they send it only to panelists or to the wider group. So you can check the union's fault. In any case, we're keeping track of it all, so it'll be there. Okay, I think we have about 20 minutes left. We've had a really good discussion. I want to kind of open it up maybe to see if we can get more people who are here to speak if they have a question, especially since there may be some anxiety about whether the written questions are getting seen or not. So we have one hand raised. I'm going to try to unmute you so that you can ask your question. And please make it a question and not merely a comment. We would like some kind of question attached to it. And so this person is on my screen as JMU. So I'm unmuting you now, and I think you should be able to speak.
Good evening.
Yes, we can hear you.
Yeah, I really commend the effort of the research team. My question will be on MDR-TB. Those people, the remaining percentage of people that the treatment was unsuccessful using rifampicin and the moxifloxacin, were they further test for MDR-TB? Is there any test carried out on them to determine whether they are drug-resistant case? Thank you.
Yeah, I can try to tackle that one. So thanks very much for that question. Just to recap that, as Dick pointed out, if you had resistance at baseline at the time when you were being enrolled to rifampin, you were obviously not included, and that was done through expert and other techniques. And the same is true for the analysis, fluoroquinolones, if that was detected early on. For those who failed to failed to achieve cure or had recurrence, there's work going on right now doing whole genome sequencing, sophisticated techniques to try to determine whether the second isolate is the same as the original one that caused disease in a patient, or were they unfortunate and got reinfected in the community with a new isolate, which does happen. And there's more work underway to find out, what the acquisition of resistance risk was. Based on interim data, very early data, we haven't had all the whole genome sequencing completed yet. It's ongoing, but based on the interim data, there is no indication of increased risk of acquisition of resistance with this regimen.
Thank you.
Okay, I think the next question, I'm going to go to my tag colleague, Lindsay McKenna, who put something into the chat a while ago. She's asking if somebody can comment on whether the regimen can be safely used during pregnancy. And I guess as a part of that question, how did the trial handle like on-study pregnancies for participants who may have become pregnant while participating in the study? Anyone want to talk about the pregnancy issue?
I can perhaps just again chime in. So at the time point of randomization or screening, if you will, for the trial, a pregnancy test was required for the appropriate patient, and it was an exclusion criteria. And so if you're pregnant, you were excluded from the study. With that said, as has been reported in many clinical trials in the last recent memory, during the conduct of the trial, people become pregnant. And we had 32 pregnancies, and they were balanced across all the arms. And once becoming pregnant, the protocol was written that they would be excluded from the experimental, basically from the trial. Would be excluded from a trial. So they would have to go to a standard accepted therapy in that local setting that would be appropriate for a patient who was pregnant. Now, remember, we're receiving these results in 2020. The protocol, and the design features were discussed, five years ago. Actually, a lot has changed in the last 5 years. I think the TB therapeutics community has appreciated much more in just the last 5 years the critical importance of gathering data on populations that usually have been excluded from trials, including pregnant women, lactating, and children. And our trial, we tried to incorporate adolescents, lower CD4 counts for HIV, but had not had the I think the consensus at the time, the general agreement at the time, to take that bold step of including pregnant women into the randomization and trial. I hope in the future that there are in fact techniques to include pregnant women in trials, and it's work that the WHO is also exploring through some technical consultations on trial designs for TB and pregnancy and children. And I hope that in the future there can be more of the kind of special populations included in studies.
And Laya in the chat has pointed out that this is something that the CRAG has started to, call for, as you just described, Payam. So I'm wondering if maybe Cynthia or Barbara or Doro want to talk about some of the work the CRAG has done to kind of push for more research in pregnant women and for their inclusion into TB trials. Any of you can start.
Well, I guess I'll speak up. And much of it is really the work that was done along with Lindsay, who's a pretty active member of our CRAG. And she has worked really hard to do the organizational work, the writing, to help move this forward. And I think I don't know if you could unmute her, because I think she could best identify where we are operationally with an official PREGLAC working group now that is a global working group, and I think she's handed it off a bit. But I think that now pregnancy will have to be included in a trial unless there's a reason that can be given why it couldn't be, as opposed to we automatically excluded. And I don't know if I'm right with this, Payam, in the next trials going forward, but I think we'll have to have a reason why we exclude pregnant and lactating women instead of vice versa.
So Lindsay has pointed out that she's not the only expert on this on the line, and I think the US TB activist Kate O'Brien is here as well. So okay. Lindsay and Kate, I don't want to put you guys on the spot if you don't want to be. So if you would like to say something, raise your hand in Zoom and I'll unmute you. But no pressure if you would rather not. While you're thinking about that, I want to take another question that's come in the chat, and then we'll return to the pregnancy issue if Lindsay and Kate want to say something. This is kind of an interesting question for either Dick or Payam. It's from Katerina Neward, and she's asking, are the number of patients experiencing at least one grade 3 to 5 adverse event still lower in the rifapentine moxi arm if you compare with the frequency of adverse events observed just in the first 4 months of the standard of care arm? Would either of you like to answer that?
I can answer it because I was just looking at it right before this call. And the answer is at 4 months, there are fewer adverse reactions in the rifapentine only arm and more adverse reactions in the rifapentine moxi arm than in the control arm. The control arm's in the middle. It's not a big difference. It's a couple of percentage points. And then at the end of 6 months, there are more adverse reactions in the rifapentine moxi arm, but they're not treatment related. And there are as many in the control arm at 6 months as there were in the rifapentine and moxi arm at 4 months. So I hope that's not too jumbled, but so adverse events happen throughout the trial and they're not always related. The relatedness of the related adverse events, things that were felt to be related to the medications, were very similar across the board. But the overall adverse events are more for the control arm because they have more time to accumulate them. I think I got that right. Sound right, Payam?
Yeah, that sounds great, Dick. And I would say in Katya, Dr. Krobatova's presentation that's on demand, and you can look at it again, she shows the sort of safety profile of liver enzyme abnormalities across the 3 regimens: control, rifapentine only, and rifapentine moxi, and looking at it at a variety of different ways of the liver enzymes being elevated. I was very reassured to see that actually the rifapentine moxifloxacin containing regimen had lower proportions of abnormal liver enzymes than the control. Now, it's not statistically significant. Well, I would call that comparable, but in terms of a consistent signal, it's lower. It's lower, and rifapentine is the lowest. So all this anxiety that many of us have carried, I think rightfully, about whether or not we could increase the dose of rifamycins to help shorten treatment, and whether we'd be sort of facing drastic liver enzyme abnormalities, is this trial is proving that's not the case, that in fact liver effects appear to be lower on the rifapentine moxi arm as compared to control.
Thanks, both of you, for that. I think that's really clear. There's another question from Aaron, which I think, Dick, this one is probably for you because it involves TPT. Payam and others, feel free to chime in as well. Asking about the implications for kind of post-TB treatment, TB preventive therapy. This recommendation that people who recently complete TB treatment for active disease should take TB preventive therapy, particularly if they live in a high TB burden or high transmission area. I don't actually personally know how often this is done in practice, but Dick, if you have any thoughts on kind of the role of post-treatment prophylaxis with this regimen.
Sure.
Note that rifapentine is part of both now. Yeah.
Yeah. So I just sent Arnie an answer in the chat, in the question, but I'll expand on it. So post TB treatment preventive therapy is really not recommended per se anywhere. What is recommended by the WHO for TB preventive therapy for people living with HIV is that everyone should get it, even if they've had TB in the past. But that's different than, post-treatment, you should get preventive therapy. And I think that is more of an operational thing, that the guidelines for people with HIV were made to make it as inclusive as possible, just a blanket across the board. Don't do testing, don't do tuberculin testing, don't do quantiferon, just give TB preventive therapy. And if they've already had TB, so what? Give it to them. There's only been 2 studies that looked at post-treatment preventive therapy for people living with HIV, and they were done in the pre-ART era. And it's clear that in the ART era, those results are not credible. If you don't get ART, post-treatment preventive therapy for people living with HIV makes sense. For Arnie, the one special case is the Western Cape of South Africa, where I know he happens to live. And TB recurrence rates there are unusually high, higher than anywhere else on Earth. And so there's been some mathematical modeling looking at whether preventive therapy would be effective there. But it's not based on trial data. It's not based on actually seeing if it works. It's just based on a mathematical model, assuming that it might work. And the mathematical model that's been published concludes that it's something that should be studied as opposed to something that should be implemented. So that's my general comment on that. Great.
So we have 5 minutes left. We need to wrap up. This has been really a rich discussion and great participation. As a final kind of word, I want to hear from each of the panelists. And what I'm going to ask you to do, and I know I didn't tell you all I was going to do this beforehand, so I'm kind of putting you on the spot, but I would like to hear from each of you your top single number one, so just give me one, advocacy issue, objective priority in relation to the study result or the rifapentine moxi regimen for the next year. What's at the top of your list for what we need to do, whether it's additional research, drug registration, price work, whatever it is, your number one issue. In terms of who's going to go first, why don't we start? Let's start with the primary investigators and we'll end with the community voices to send us off. So Payam, as the investigator from the lead network for this study, why don't you start? And I'm going to take notes on these because I think this is our agenda for the next year to work on together. So I'll be writing it down and Payam, over to you.
Thanks, Mike. No, I'm actually going to draw from Ken Castro's comment in the Q&A. I think the chief advocacy work really is about seizing this moment and taking these results and taking them forward in a setting where we're all living in a pandemic that's on top of an epidemic and another epidemic. So this is time for action. It has always been time for action. It's the advocacy and civil society that can help us accelerate that. You asked me for one, I guess that would be it, because I think that will generate the energy needed to move from a successful trial outcome to all the other steps of the race. This is a marathon, and we've only just reached the first leg of the race where we've finished first place with this rifapentine moxifloxacin, making a 4-month regimen that's robust for everybody. But there's a lot more work to do. TB is an infection. It deserves diagnostics. That needs advocacy. It's a disease that needs multiple drugs in a regimen, and that needs advocacy for making pill burden less. It needs access in resource-limited, impoverished settings. So all those things fall into place, but that would be my summary.
Great. Dick, over to you.
So we have a bird in the hand, so to speak, and we shouldn't wait for something better to come along. We should work for something better to come along, but we have a bird in the hand that's a big, huge improvement. And so in the next year, I would like to see it adopted as a recommendation by the WHO, by the CDC, ATS, IDSA, and put into practice. And within the year, I'd like to see serious movement towards fixed dose combinations or better products. More accessible, but better products. And I won't be any more delusional than that.
Good. That's enough as it is probably. Doro, what about you?
I have quite a number, but since you asked for one, I would say that oh my God, I'm trying to see which one I could prioritize. But for the next year, which is in the next 2-3 months to come, I think one of the things that we will need to focus on is ensuring that we will need to have more these results disseminated. Dissemination of the results is important, but also ensuring that the WHO goes beyond just the statement, but also having these maybe translated into guidance on how we can, how this can be adapted into guidelines, new guidelines. We need new guidance from WHO guidelines. Yeah, having new guidelines from WHO. And if we can start from there, then we see how that translates into our countries. And we look at prices. What is the cost? The cost of that, because that is another issue that we need to look at. Because accessibility looks at also pricing.
Awesome. Great. Cynthia.
Yes, so I guess, I mean, we have so many asks, but for me, looking at the work that the scientists do, the number one priority that I could ask is more funding for TB research. I don't think it's fair that we have such great scientists and they always have to just make do with what they have. And it's like every disease that comes along gets more funding than what TB gets. To me, that's priority, to make sure that our researchers have all the resources so they can focus on research. And definitely we would make so much headway with TB treatment if they had the money available. But I think they do research within the means that the funding dictates. And I think that's very unacceptable. So that's my priority.
Yeah, I love that. And Barbara, last but not least.
So I think I feel really strongly about this. One of the key ethical principles of research that all, at least researchers in the US, need to learn and regurgitate back on an exam before you can do research is that communities that bear the brunt and the risk of research should reap the benefits of the research. So I think we need to work very hard to implement this very powerful study, because how else do we justify moving forward with the next set of TBTC or other trials? Because the drugs are going to be, in general, more expensive, more difficult to implement. These are drugs we know how to use in our TB clinics. And so personally, I'm sitting here in San Antonio, which is one of the trial sites, and I have already talked with my colleague, Dr. Lisa Armitage, and we're going to work hard to implement this into our TB clinic here in San Antonio, and hopefully follow the patients and see how they do and monitor them very carefully for, tolerance and what happens with this. But I'm going to commit to helping Doro, and I think we should do this as TAG and TBTC and Craig. This is a community, her Uganda community, really bore much of the brunt of this research, and I think should be one of the first places in the developing and lower income countries, middle and lower income countries, to reap the benefits of the results of this trial, because they have borne the brunt of so much research and Doro has worked so hard. So that would be my goal, with 100 other things I want you to do right away, right now, tomorrow.
Yeah. That was so powerfully said. I'm so glad we ended with you, Barbara. Communities that bear the risks of research deserve to reap the rewards of research. And as CRAG, we've always said that access should start with the countries and places that contribute to the clinical trial and make it possible. So that's where our group will be starting our work, and we'll invite everyone to join us from their locations, wherever those may be. We need to wrap up. This has been really great. I want to thank all of our panelists for giving us so much of their time today, to say congratulations again to CDC, to NIH, to the CRAG, to the NIH community mechanisms, which have all just been incredible. It's been a 40-year road to get here. And fortunately, as Payam reminded us, we're just halfway through the marathon. There's much more work to do. So we can't get tired, but I think we can celebrate a little bit and then get down to work. So that's it for us. We're going to sign off. Goodbye to everyone. Thank you so much for joining us, all of the attendees, and for your participation. And I hope you have a great rest of the conference, and get in touch anytime, and you'll be hearing from us on this, I'm sure. So bye, and thank you.
Thank you, Mike.
Thanks,
Thanks,
everyone.
Mike. Thanks.
Great session,
Thanks
Mike.
all.
Bye,
Bye, everyone.
Bye.
Bye, Doro.
Thank you, Mike. Thank you, everyone.
Thanks, Goro.
Bye.
Bye.